Okay, so we're going to look at the cell cycle. So here's your textbook reference. In your bio books, you'll find the content relating to what I'm going to talk about here on pages 358 to 362. So for this page here, what you'll see are the learning goals on the top right. So you have to be able to name the main phases of the cell cycle and also give a description of what happens in each of these phases. So there are 3 phases. It's important that we can break each of these down. So how about we start with that? Now, how do we name and diagram the 3 phases? So the 3 phases that are given are interphase. And this is the order that they occur in for a cell, by the way. Interphase starts off the cell cycle, and it's the longest phase. This is really the longest phase. So if a cell took 24 hours to divide, interphase would be like 20 hours of that cycle. Mitosis is the second phase, a highly coordinated process. And then we have cytokinesis. So these are the 3 phases. Now I'm going to draw. I find it harder to draw a circle on my computer than it is to draw on the board. Okay, so we have here the cell cycle. And most of the time spent by the cell here is interphase. This section here is mitosis. And this final stage here, where the cell divides, is cytokinesis. So this is the cell cycle going clockwise, okay? Oops. This is the cell cycle going clockwise. So we go this way. Okay, so cells just continuously cycle through this. So we need to know the order and roughly, I guess, the proportions of each of the phases. Okay, so what happens in each of these phases? Let's start with interphase. What happens in interphase? Now, interphase can be broken up into 3 subphases. And the 3 subphases are G1, S, and G2. G1 we call gap 1. I like to think of it as growth 1, because what is marked by the G phases, is growth. All right, so growth happens in each of the G phases here. So with gap 1 and also with gap 2, we have growth. But more specifically, what happens in each of the gap phases varies. So with the gap 1 phase, what we have, well, with each of the gap phases, we see that the cell increases in size. And develops its organelles. So it replicates its organelles and so on. But it prepares for the next phase. So it prepares for the S phase. So we kind of need to know what happens in the S phase. Now the S phase is known as the synthesis phase. And the reason why it's called the synthesis phase, because what we see is the replication of replication of DNA. So basically all the chromosomes, they double. So each chromosome consists of one chromatid. So I'll say chromosomes start off as single chromatids. And then by the end of S phase, each chromosome is two chromatids. So those chromatids have replicated themselves. Okay, so gap 1 prepares for synthesis by developing all the protein machinery that is required for DNA synthesis, for the replication of DNA. So it's important to make that connection. That gap 1 supports synthesis. Okay. Now gap 2 is the phase that occurs right before mitosis. So you can probably figure out what gap 2, one of gap 2's roles would be. What are one of the things that happens in gap 2? So gap 2, yes, it has like some of the typical growth things, like the cell increases in size and replicates its organelles, but it prepares for mitosis. And the way that it prepares for mitosis is that it develops some of the machinery, the cellular machinery that is required for mitosis to occur. So in mitosis, which we'll look at in more detail later, we have like particular structures like this, the centrosomes that are involved with pulling apart the chromosomes and so on. These need to be replicated. So in gap 2, this happens. So yes, gap 2, we have like the usual stuff up here, that the cell increases in size and develops its organelles, but it also really prepares for mitosis. So make sure that we can establish these key features of these subphases. All right. So we've deconstructed interphase. Now with mitosis, you really need to know each of the steps, the names of each of those steps, and roughly what happens in each of those. So with the first one, we have prophase. And in prophase, we have the condensing of chromosomes. So the chromosomes become really tightly packed. This is what condensing is. Condensing of chromosomes. So if you look at diagrams of prophase in your textbook, you will find that like the chromosomes are like kind of really, not really visible yet because they're just a massive ball of DNA. But then by the end of prophase, they're really, really clear because they've condensed into very thick structures. So in prophase, we have this. We have the condensing of chromosomes. We also have the breaking down of the nuclear envelope, among other things. But I think here we'll just talk about what happens to the chromosomes. So we have the condensing of the chromosomes, where we find the DNA. Then in metaphase, we have the chromosomes arranging themselves. Arranging themselves. And so they arrange themselves in a very particular way. They line themselves up in the middle of the cell at the location we call the metaphase plate. And they do this so that the chromosomes can later be distributed evenly between the two ends of the cell, which then become the two new cells. So the chromosomes arrange themselves in metaphase. Then anaphase, they actually separate. So chromosomes separate. And then telophase is really, I like to think of it as like the very opposite of prophase. So in prophase, we see the condensing of chromosomes and the breakdown of the nuclear envelope. In telophase, we see the reforming of the nuclear envelope and then the decondensing of chromosomes. So by this stage in telophase, we will be getting like pretty much 2 new nuclei in the cell that's about to divide. And these 2 new nuclei have an identical set of chromosomes. So they have pretty much the exact same DNA. And so what follows mitosis is cytokinesis. So cytokinesis is the splitting of the parent cell into 2 new daughter cells. So from mitosis, we had the distribution of DNA into 2 new nuclei, and in cytokinesis we have that cell with the 2 nuclei being split. So we get 2 cells with identical DNA. Okay, so these here are the nuclei. So nuclei with identical DNA. Okay. So these are the phases of the cell cycle. And we did go over it very quickly, so of course, if you, and you should be doing this, you should go into your textbooks and read in more detail what happens in each of these phases and the subphases. Okay.